Thank you very much. We apologize for the modest technical difficulties this morning. Welcome everyone and season's greetings. And I hope that that lump at the end of your driveway did turn out to be your car when you dug out. For me, it was. So good luck. This has been a very impressive 2 days of snow here in the Schenectady area. I'm excited to be back with you again. This is my third presentation on this interesting virus and pandemic that we're experiencing right now. I'm going to go back a little bit, not too far, as we go forward to make sure that we close some loops and close some items that we've talked about in the past. Just to let you know where this aircraft is headed. First, I'm going to have a quick look at influenza. I just couldn't resist. Remember, my October talk was about whether we thought we would see double trouble. And I have some data to show you about that. Talk a little bit about the current and a quick snapshot of the status of this epidemic and pandemic. Talk a little bit about the fatigue we're all feeling, not too much, but we're all feeling that fatigue. And then also the danger, potential danger of seeing the vaccine and thinking we're home free, which of course we're not yet. Talk briefly about a history of vaccines and what they've done for public health. Talk a little bit about how vaccines have been made in history and what the usual development of vaccine looks like in the current moment. Then why the mRNA vaccines are revolutionary. And please keep in mind, those listening to the call and watching today, that we've never, ever had an mRNA vaccine widely deployed and distributed. So what you're witnessing, what we all are witnessing, has been created in the past 9 months is brand new technology. Very exciting, extraordinary in many ways, and I'll be effusive and talk more about that as we go on. We're going to then talk about which the candidate vaccines have been, and there have been a huge number that have been, work has been begun on. Then we'll talk about the funnel-like effect and kind of where we are today with some candidate vaccines that are either going to be deployed and have been deployed or perhaps will be deployed within the next month or so. A question that we all are asking ourselves, when does this end? What will the end of this pandemic look like? And then I'm going to close with some frequently asked questions and a summary. Several of you have written to me knowing that I was going to present today with some ideas, excellent ideas. I've tried to incorporate your requests into some of the calculations that I've made and some of the material I'll present. So thank you for that input. One small note here, declaration, you need to know that I am in fact a Moderna shareholder, and that happened actually in January. And it was more that I was in love with the technology than understanding that they would actually build a successful vaccine. Having said that, I would encourage you to take any vaccine that has an emergency use authorization provided by the FDA. I am indifferent to what you take or what I take. So please know that although I am financially engaged with Moderna, I'm indifferent about how you get immunity. So please, if it's offered, take it. Now, going backwards just a little bit, we talked in the October presentation about whether this would be a double trouble winter. That is, would we see seasonal influenza coincide and collide with the COVID-19 virus? And I think that what we see, and if you can look down here at these diamonds, these triangles, I should say, the red triangles, those are the influenza-like illness reports from the surveillance network that is established by the CDC. What you're seeing here is very low activity, as low perhaps as the season of 2015, 2016. We don't know where this is going to go because remember that it's usually right at the end of the year when we begin to see these very large spikes. But right now, our experience is looking more like Chile and more like South Africa and Australia during their last winter, which of course was our summer. So right now, a little bit of cheery news that we may not see double trouble, and in fact, very low activity from seasonal influenza. I haven't drilled down to look at the serotypes, but eventually I will do that. And if there's anything important, we can always talk about that maybe in the winter. When we spoke last, that curve that's on your left, the one that's in blue, the arrow that's pointing there is to what October looked like. And remember, I said, we're not sure whether this is going to look like a blunted wave or whatever. And as you all know, and we all know, that wave has turned into a tsunami. We're just seeing a density of wild virus activities in our communities that is very, very impressive and, of course, very dangerous. And you're all aware of the fact that in America, we've had roughly 17 million reported cases, culture-proven reported cases of this virus. And most epidemiologists and virologists would tell you that is certain. More than 350,000 deaths and about 3,500 fatalities occurring daily. These are stunning figures. Unfortunately, the coldness of the numbers never relays the depth of the sadness that goes around these numbers. But there are families suffering and individuals suffering and healthcare professionals suffering under the burden of this disease every single day. So thank you to frontline workers who are on the call today, and thank you to frontline workers who will be listening to this sometime in the future. I hate to be a complete Debbie Downer after telling you we probably won't see much flu, but I'm inclined to believe, as other experts have said, that we are probably going to see an increased density of illness after the holidays because of the desire, our human desire for social contact, and the fact that we'll be putting people together who unknowingly, unwittingly are asymptomatic and carry this virus. So I would have to say we have to brace ourselves and certainly be socially highly responsible with our own behavior. And in fact, healthcare professionals, all of us, everyone listening and watching today, hand washing, wearing a mask, social distancing, avoiding even small familial crowding, all are critical elements to trying to blunt as much of this curve as we can. Some of this is already loaded in the population. That is to say, there's so much virus that we can certainly anticipate hospitalizations and, of course, as a consequence of hospitalizations, fatalities. So this is going to be tough. It'll be tough probably for the next 2 months. We'll have to see, but at least it's going to be tough for 6 or 8 weeks. So now to talk a little bit about the background of vaccines and what we've learned and where we've come from. The first slide, the one that now shows diphtheria, polio, and measles on the left-hand screen, simply shows with the red dot when the vaccines were introduced. And you can see the precipitous decline in the cases of diphtheria, polio, and measles after the introduction of those vaccines. And on the right-hand side, whooping cough and the marked reduction in cases of whooping cough. Only clean water and sanitation have had a more profound impact in the health of human beings than vaccines. So vaccines is right up there in the pantheon of great achievements in public health and in all of human health. Vaccines, in fact, work. Their impact on our health has been profound. Those who are anti-vaxxers and vaccine deniers are living and occupying their own world. The evidence is unequivocal. These vaccines do work, and we should all encourage ourselves, family members, and others to make sure they follow guidelines, CDC guidelines, and getting immunized at the proper time with the proper vaccines. Pardon me. So when we think about vaccines, and in the prior slide, you saw there are a number of different kinds of vaccines that have been created over time. There are ones that take the virus and grow it in numerous cycles to weaken it or attenuate it. We have seen some success with tuberculosis in this with BCG, although BCG's impact in the West is limited In developing countries, it's been a little bit more helpful. Oral polio vaccine, that which was developed by Albert Sabin in the 50s, was miraculous and wonderful. We have now gone back to a killed vaccine, but initially Albert Sabin's contribution made a profound impact. Measles, rotavirus, which the pediatric folks and family medicine people will be familiar with, and then yellow fever, for those of you who have been global travelers, all are part of this live attenuated way of thinking. There's also, of course, a live attenuated influenza vaccine, which has been of marginal value over time. Inactivated vaccines include things like original polio vaccine that was created by Jonas Salk and his team in Pittsburgh. A profound impact on my childhood. I was a kid when the polio epidemics were sweeping through the country, and we had similar limitations to what are asked for today. We couldn't go to pools, we couldn't go to movie theaters. There was very little social mixing at all during those summertime waves of infantile paralysis. This was largely a disease. Polio was a disease of children, causing paralysis and death in some individuals. The middle is a subunit kinds of subunit vaccines, including pertussis vaccine that is free from the capsid that goes around the pertussis bacterium, influenza, H flu or HIB, the pneumococcal, moiety, and Hep B, and then toxoid vaccines like diphtheria and tetanus toxoid. And then on the extreme right, next generation in this particular chart, are the mRNA vaccines, and we're going to talk about those in the future. This particular slide divides them into replicating and non-replicating moieties. We'll talk about what we're dealing with in this particular time in this pandemic. So there have been notable, glorious successes of vaccine development. I couldn't help myself on this slide either, including smallpox, which I was privileged to work on in India. Yellow fever, rabies, DPT, MMR, meningitis, HIV, Hep A and B. So glorious and notable successes. There have also been failures. So as we hope for the benefits of the vaccines that are coming down the pipeline, we have to remember that we've not done so well with Lyme disease, HIV, AIDS, tuberculosis in a more grand way, dengue fever, hepatitis C, malaria, and influenza. Which, yeah, we do have some vaccines, but I think none of us are satisfied with the protective value, the vaccine efficacy of the influenza family. There is a hope, and if time allows, we might talk about this. There's a hope that we might be able to use an mRNA vaccine to make influenza a better vaccine and perhaps make it so we don't need to reformulate it annually. That's a dream. And maybe a pipe dream, but at least there is a possibility that by using mRNA technology, we could advance the influenza offerings that we give every year. So this slide takes you to the traditional way vaccines like MMR, like some of the other ones that have come on the market, meningitis and others. It begins with an idea, a dream, a target illness that affects a lot of people, has economic consequences, and is devastating. So once the target is found, then preclinical work is begun with animal models. And then once an animal model is successfully designed and provides convincing results, then there are 3 phases of vaccine development. An initial one just to make sure that it's tolerated and not going to cause serious problems. The second phase then is ranging often, giving multiple doses, and again, making sure the untoward effects aren't bad, and that in fact, it triggers an immune response that's robust and valuable. And then finally, phase 3, where these cohorts of 30 or 40,000 people are enrolled, immunized, and then controlled with a placebo, then followed forward in time in the face of wild virus, in the case of the mRNA vaccine we're talking about today, and looking at the 2 populations. What was the rate of infection, so-called attack rate, in the people who were unimmunized versus the attack rate in the people who are immunized? From that, we can calculate vaccine efficacy, and we can also calculate a number that we call number needed to treat. That is, how many people would we need to present with a safe, effective vaccine to prevent one illness? And from that, you can extrapolate how many doses you would need to give to prevent one death. We'll talk about that in a few slides. Once you've gone through these trials, you need to go to the FDA for approval and then manufacturing. Now, it's important for the people listening today and watching to realize that what's happened for these two vaccines, first, the one that is the BioNTech vaccine with Pfizer and the Moderna vaccine, they have not been FDA approved. They have received emergency use authorization, which is different. It's similar to the way what's happened with remdesivir. It's similar to what has happened with the monoclonal antibodies that we use for this illness. We have authorization, but not the rigorous approval that usually goes on. So you need to realize that some of the length of time that has been used to evaluate other vaccines has been shortened slightly for these vaccines, but rigorous nonetheless. And then finally, manufacturing. Now, if you look below, there potentially are some ways to short circuit this process. That is to say that if you get a vaccine in a phase 2 trial that begins to look very good, you can go to the NIH, you could go to the federal government and say, this looks really good. What we'd like to begin to do is begin to package this and build doses. And you'll see in a second, that's exactly what happened with the mRNA vaccines. We simultaneously were doing the field work and experiments while we were packaging and filling vials with an mRNA vaccine. Here is the timeline for the Moderna product. A similar timeline was used with Pfizer and BioNTech. The BioNTech people are a German company. They began to see they were having early success with their product. They called Pfizer on the telephone and said, we've been working together. I think we should team up and really begin to build volume and run large-scale studies. And that's exactly what they did. Now, I show this and circle this area on the timeline to talk about how extraordinary this was. You'll remember that when the outbreak began in Wuhan, in China, the Chinese very quickly sequenced the virus, the genome of the virus. They then published that on the web. The sequence of the virus was then available to any researcher, any place on Earth to do what they want with it. So both Moderna and the BioNTech people in Germany grabbed the virus and they began to build mRNA segments like the spike protein. Now we've learned that the spike protein that's gone into the vaccines is a slight variation on the actual natural spike protein, because we've learned that when the coronavirus attacks our cells in a lock and key fashion, in fact, it modifies the spike protein slightly when it resides in our cell. So if you harvest the protein from the cells of mammalians, what you find is a slight variation on what you really need to get at. So a minor tweak and some base pairs have been made in the mRNA for these vaccines, and as you'll see, has turned out to be very beneficial. But what I want to bring your attention to is that from the time of receiving the sequence from the researchers in Wuhan until putting a dose of vaccine in a human was 63 days. 63 days is, it's amazing. It's stunning, and it's like moving at light speed versus a bicycle. So they went from a genomic sequence to a vaccine in just a shade over 2 months. Ordinarily, it would take you 4 or 5 years. So the breathtaking speed with which this has happened, for those of us who do vaccines, infectious disease, and public health, is stunning. I mean, our heads are still spinning to think that this occurred. So as soon as the phase 1 work was in and the vaccine looked to be reactogenic to a degree that was completely tolerable, the people at both Pfizer and Moderna were told, begin packaging, manufacture and package your product. And as you know, last week we had the Pfizer BioNTech vaccine that was authorized, and yesterday and today, the Moderna product also authorized. So for those of you who haven't thought about this or conceived about it, I think it's comparable to putting a man on the moon. Now, you may think that I'm being hysterical, but in fact, it has taken more than 10 years for the mRNA vaccines to be conceived and packaged and so forth. We know that Moderna, and we also know that BioPharma have a number of vaccine candidates they've already built using this technology. They have animal studies to support the fact that it's worked, but not without successive failures over at least a decade. And the real trick has turned out to be encapsulation, that is getting the mRNA inside a lipid nanoparticle so that it's not destroyed by our innate immune system, so that it gets into muscle cells and other antigen presenting cells and is able to get to our immune system so we replicate antibody to it. It is extremely exciting, and millions of doses of this vaccine will be shipped and will be put into the arms of Americans very soon. But those of us who live near the sea recognize those flags on the left as being hurricane warnings. And the high wire man on the right, sadly, the World Trade Center, reminds us that we're in a very, very precarious period. You know, it's ironic that as the vaccines arrive on the sea, we are entering probably the most calamitous period of this pandemic right now. So there's no letting our guard down on the routine measures that we know mitigate transmission, illness, hospitalization, and death. So please do not be lulled in to the siren song of, oh, the vaccine is here, you know, we're in the clear. We are not in the clear. We've got a long way to go. Now, just to move from the sublime to the kind of nuts and bolts, several groups have tried to look at the coronavirus in different ways. Maybe we could make an RNA vaccine, which we're celebrating today. Maybe we could attenuate this virus like we did with measles, and we also did with the whole entire MMR and chickenpox. In fact, a group in India is working on that, and a few other manufacturers as well. The Chinese have a product that they're working on. Inactivated, the inactivation of a virus, you simply take batches of viruses in vats and you kill the virus and you make sure that it is dead when it comes out of the end of the vat, and you inject that into subjects so that they will produce antibody against the elements of the killed virus. Now, that worked very well against polio. Polio, which is an enteric, that is a GI virus that causes paralysis. Thank goodness most of you have never seen a case, but polio was a devastating illness, and this worked very well. Then there are subunit vaccines like whooping cough and Hep B, and then there are viral vector vaccines like the one that is being used at the University of Oxford with AstraZeneca and Johnson & Johnson. This is using usually an adenovirus, and you splice in the sequence of genes that you want replicated, and the cell itself begins to replicate these elements, including the antigen that you're most interested in this case, the spike protein from the outside of the coronavirus. When the gun went off and people began to decide could they like to try to make a vaccine, there were 154 vaccine candidates in lab experiments in animals that were started. And as many of you have read in the popular press and heard on TV, a great number of biotech firms dropped everything else. They stopped all the other work they were doing, and they turned their teams exclusively on to developing vaccines against this particular terrible, devastating pandemic. And so we winnowed it down from 21 to 13 to 10, and we're going to talk about the more winnowing that's gone on. No approved vaccines yet, but 2 vaccines authorized. So the offerings and the participation in clinical trials have begun with the J&J product. Now, J&J, Johnson & Johnson, holds the hope of only needing to give a single dose. There are rumblings inside the vaccine world that they're not getting the seroconversion rates they want, and they may, in fact, begin running a parallel trial with 2 doses. AstraZeneca, you've heard, they had some dosing issues. They've had a few other issues with their product, the Oxford product, but they're coming along. I don't know much about the Novavax subunit vaccine that's in trials, but we've already talked about, I'm just going to make a note there, sorry. Then the Pfizer product we've talked about, Moderna we've talked about. Sinovac is a Chinese product, and that the Sinovac vaccine there is inactivated, much like the early polio vaccines. And the Sputnik vaccine made at the Gamaleya Institute in Moscow is a viral vector vaccine. We have not heard much about that in terms of what the data show. As you know, even before approval, the Russians decided to make, package and deploy that vaccine. And I guess apparently Mr. Putin's daughter received that vaccine before the data was really carefully scrutinized. Fortunately, that's not the case with the vaccines that we have available here in the
full screen for a minute and try to go back in. There you go. Okay.
Okay, sorry. So here we go then. Pardon me for that technological glitch. So this is just what Moderna put in their emergency use authorization application. This is the statement they made. And so I think the key thing here for all of us is just to be aware of the fact that the mRNA vaccines, and this goes for both Pfizer and the Moderna product, these only enter the cytoplasm. It doesn't go into the nucleus. It's non-replicating. And it is only expressed transiently. In other words, it goes in, replicates, it appears in the cell surface, your immune system see it and begin to create antibody to it. I love this analogy. One of the science writers said it's really a little bit like an M&M. You know, the outer coat is sugar, protects it, and the inner coat is where the business is, that is chocolate. In this case, you can see we have this lipid nanoparticle that's carrying inside it the messenger RNA, which gets into the ribosome, and the ribosome then recognizes and says, wait a second, I see this code. I know what this means. I'm going to make a protein. And so it begins to make the appropriate spike protein, which then sits on the cell surface, which then rings a bell and says to the immune system, hey, there's something funny here. I think we probably ought to make some antibody to that. So this is a simple kind of rock and roll version of exactly what goes on. There's a much more elaborate business goes on with lymphocytes and in lymph nodes. But suffice it to say, think of M&Ms when you think about how this vaccine gets into our cells and then how our immune system recognizes the protein that is commanded to be created by the mRNA when it goes through our ribosomes. This is just to let you know some of the nuts and bolts about the vaccines. And you can see here some of the characteristics of the way it's got to be held, the effectiveness that has been reported to date. As some of you are aware, the Pfizer BioNTech needs to be kept at ultra cold temperatures. Moderna cold, but cold like most of us in our offices are used to. The people at Ellis have done a marvelous job dealing with the ultra cold thing, thawing, reconstituting this vaccine and putting it in your arms. So it can work very, very well. So I think that's a technological hiccup that is easily dealt with in folks like Ellis. Now, in rural communities, that could be more of an obstacle, but for right now, we seem to be doing this pretty well. So just getting into some nuts and bolts, we talked about ultra cold storage and shipping. After 5 days, the Pfizer product can be held in fridge temperatures. The Pfizer company provides you with multi-dose vials. They need to be thawed, and then you have to add a diluent, 1.8 milliliters of diluent. And the reason I specify that is because if you divide 1.8 by 0.3, which is the individual dose, you very quickly come up with 6 doses per vial, even though the company promised you 5. So 0.3 is for intramuscular delivery. The upper arm is preferred, like you saw on TV. And within 5 hours of reconstitution, you really got to use the product. And then after 6 hours, probably you should discard it, which means that we want to make sure that no dose goes unused, right? This is precious stuff. Make sure we use it. The contraindications, as you've heard or perhaps read, severe allergic reactions to any of the vaccine components, and even people who've had prior anaphylaxis need to be aware and wary perhaps of using this. Now, I know, for instance, that allergists will immunize people with egg allergy to certain of the influenza vaccines. We may get to a point where that might be possible in the future, but for now, we believe this is a contraindication. The Moderna vaccine is a frozen suspension. It's kept cold for shipping and storage from minus 15 degrees to 5 degrees Fahrenheit. They are multi-dose vials containing 10 doses of 100 micrograms per dose, and there is no dilution. So you do not need to enter the vials with a syringe and a needle to add the diluting solution. You simply need to begin drawing doses once it's thawed. You can hold it in refrigerator temperature for 30 days, and it can tolerate standing out in a clinic between 46 and 55 degrees for up to 12 hours. Again, IM goes into the muscle, but this is a 5 milliliter delivery. This is 0.5 mLs. So it's a different amount, same location, different handling of the product itself. And once again, I think if there have been prior severe allergic reactions, one needs to be aware of that. In addition, some of you have heard there's been a facial paralysis associated with some of the Moderna vaccine recipients. It appears to be an acceptable frequency. Most of this facial paralysis is self-resolving, and we did see this palsy called Bell's palsy in the placebo controls as well. So after 21 days, we boost the Pfizer product, and after 28 days, we boost the Moderna product. So dose day 1, dose 28 or 21 or 28, depending on which. The nuts and bolts of this are extraordinary. I've been on phone calls with the COVID vaccine group at Ellis, and the details and tracking and electronic medical record and insurance and cards and giving you vaccine information sheets, getting you registered, getting you inoculated, observing you for 15 minutes, handing you cards with reminders. This is a big deal. I mean, this is an enormous logistic thing. And we have the nerve to think we're going to be able to do this with, I don't know, 300 million Americans. So very impressive. So as impressive as developing the vaccine is, the deployment and logistics of that deployment is equally impressive. Now, of course, ethicists and CDC and the state of New York point out that we have very clear marching orders for different cohorts of individuals who will get this vaccine. They'll range from frontline providers, folks in nursing homes, people who are vulnerable populations, areas of the state of New York with high frequency of wild virus activity. There are many permutations and combinations into thinking who should get it and when. So I would urge you simply to stay tuned. Some of the practices in the capital region are already notifying patients that they're planning immunization clinics once they begin to get the vaccine. A little bit of good news is that there is some vaccine efficacy apparently after a single dose of both of these vaccines. Both of, when you do the calculations, comes out roughly 50%. Now, neither Pfizer nor Moderna will say that you really can assure yourself at a month or 2 months that you have 50% protection. But in the period of time that they studied, it appears over that roughly a month or two that you had protection at a level of 50%. I would remind the listeners, the viewers today, that 50% is roughly in the ballpark with the influenza vaccine that we give every year. And 50% was the bogey, was the target that we hoped we'd hit with any of the vaccines. So as you'll soon hear, we way exceeded the efficacy that we hoped we would have with the final products that we currently have available to Americans. The FDA application from Pfizer, and you can see greater than 7 days after dose 2, it's 95% efficacy. And remember, I told you the efficacy is simply calculated by subtracting the number of cases that were immunized from the cases that weren't immunized, and dividing it by the number of cases that weren't immunized, and then you get this number here, 94.8. And you can see there were only 50 cases of the disease, the COVID-19, in the people who received the vaccine, the BNT162b2, versus the placebo, where there were 275 cases. This is out of 21,000 individuals. So there's a large number of folks and a surprising number of cases here, 275 cases. One of the ironic benefits of being in the midst of a pandemic is that you can test vaccine efficacy more quickly when there's a great deal of wild virus activity, simply because you're able to calculate the difference between those who get it and those who don't in an efficient and very rapid manner. So when the vaccine trials began, because we have so much virus activity, you can reach a conclusion pretty quickly. This is the Moderna calculation, 94%, roughly calculated the same way. This is a series of this is a table I built, and I have one for also the Moderna product. I simply point this out because you can see that people had, this is solicited reactions. That is to say, the investigators asked the people who got the vaccine, did you have any problem at your injection site? Did you have any systemic reactions, fever, chills, achiness, pain, diarrhea, anything like that? So these are solicited reactions. So you can see more than 2/3 of people had a little bit of injection site discomfort, greatly more than the placebo. And in the second dose of vaccine, roughly the same. So more than 2/3 with some local injection site feelings. You can see the amount of systemic reaction, that is, some of these things, was greater in the second dose, similar to perhaps Shingrix, where we see the second dose causes more reactogenicity. Fever, you can see in the first dose is relatively mild, 3.7%, but when you get that second dose, it goes 5 times higher. So there's more fever with that second dose of the Pfizer product, headache similarly, and fatigue in a similar way. These reactions with the second dose has allowed certain facilities, certain hospitals, certain groups to stagger who of their staff they would present with the vaccine, simply because they're concerned that some of these untoward effects could make people need to stay home and be out of work. Here's the same table that I created. This is for the Moderna vaccine. And what's really fascinating is when you go through the table and you begin to look at the numbers in detail, you find out that even some of the manufacturers make mathematical errors. In fact, you see the number here, 62 for the frequency of headache, pretty big number. In their data tables, it's 6.2. I had to recalculate that number to recognize the fact that almost 2/3 of the people with the second dose of the Moderna product got a headache, and a lot of them got fatigue, right? And muscle aches as well. Fever, I mean, yeah, it's up there. 17% of the got the vaccine, very low number in those who did not. So what I've said, by looking at the data tables carefully myself, and these again are solicited, did you have this, did you have that, not unsolicited. But by inspection, it looks to me like the Moderna vaccine has slightly more reactogenicity with that second dose. Here, I just did some calculations to verify the fact that I got the same numbers that they got. And then I did something that's called number needed to treat. The number needed to treat is simply to 1 over the difference between the attack rate in those who were taking a placebo minus those who are getting a vaccination. So what this number says is that you'd need to immunize 114 people to prevent 1 case, right? And so also in subjects over 85, the vaccine efficacy was about 93% for Pfizer. And in order to know the number needed to prevent a death, you would probably have to multiply the number needed to treat by 100. So it's a large number of people to immunize to prevent a death, because the attack rate, fatality rates are in the 1.5%, 2%. So just if you're interested, it would be thousands to prevent one death. The Moderna vaccine, similarly, I recalculated their vaccine efficacy. I looked at their how they performed in people over age 65. Very good performance. They say 100%, never 100%, but certainly a very high number. And their number needed to treat was 80. So we need to give 80 doses of the Moderna vaccine to prevent one infection based upon the data they presented and the application. So finally, just to say that number needed to treat is important, but what goes on in vaccine deployment is that you get a dynamic. So you're giving the vaccine, illness is occurring, but the vaccine is spreading a greater and greater wave of subjects that no longer can provide a safe haven for the virus. So things begin to change. They change slowly initially, and by the time we begin to get lots of people immunized, they can change very quickly. We can see a rapid decline in what's going on with wild virus. Just to give you an idea, put the in perspective efficacy, MMR vaccine, one dose gets you 93 vaccine efficacy. MMR vaccine is 97% effective against MMR. So just to give you an idea that the efficacy that we got from these two candidate prototype mRNA vaccines, extraordinarily good. You know the CDC recommendations, healthcare personnel, residents of long-term care facilities, and then the state of New York has got an additional idea about hotspots and getting vaccine into hotspots, which makes logical sense. We've talked about the priorities, and you've no doubt already heard about that. So this is a question: how far away is the end of the tunnel? And here we're talking about herd immunity. Herd immunity has gotten such a bad name in the popular press that I want to make sure that people understand this is just a concept, okay? This is a concept that says, when will the population have sufficient immunity so that the transmission of virus is stopped, comes to a halt? That's all herd immunity is. Now, allowing the wild virus to rampage through your community is a way you can get at herd immunity, but very few groups, very few societies have taken this posture. Now, the Swedes initially did something like this, but they've come back and stepped back and realized that that probably was too great a price to pay for the burden of illness and death. So the way you get to a sense of herd immunity is you need to be thinking about three populations: those who are recovering, that had the infection, those who are currently ill because they're building antibody as they go, and the number of recipients of the vaccine, assuming it's, the efficacy is 95+%. And so you need to know that the US government has ordered 500 million doses of vaccine from various suppliers: Johnson & Johnson, Pfizer, Moderna, Novavax, AstraZeneca. And right now, we're seeing the Moderna and the Pfizer product shipped. Probably there will be 50 million people immune in a month, roughly, and maybe 6 or 8 weeks. Maybe it's going to take longer than I think. But remember that each immune person, however you become immune, denies the virus a home. So the more people we get immunized, the more we avoid letting the virus move by taking measures like social distancing, masking, hand washing, so forth. All of this buys us time to build up the immunity from the vaccine. So my guess, and this is, fearless forecast here, and it's always fraught with problems. What a Yogi Berra said, I always avoid predictions, especially when it involves the future. But here I'm speculating that it could be July, which is a long way off before we really reach herd immunity. Before that, a lot of us will be immunized, but remember, we need to get to roughly 200 million people that fall into one of these categories. So there are persisting questions, and some of you have raised these in emails with me. So are the Pfizer, BioNTech, Moderna vaccines safe and effective? And I think the answer is almost certainly yes. I think we have sufficient information that I can reassure you. When will the Moderna vaccine ship? I think it's going to be a Hanukkah Christmas present arriving next week. Is the natural immunity, that is, if I get COVID-19, will I be immune for a prolonged period of time? And the answer is, we don't know. The answer further, though, is probably because we know from experience with childhood coronaviruses of other kinds that there is probably durable immunity. We'll see, but it appears that we can have at least some confidence in that. This raises a point that has come up in emails again that I've had, is should we immunize people who have previously had PCR positive tests for the virus? And that's a tricky one. In the vaccine trials, they did do that. Ultimately, I think we will do that. But while vaccine is limited in its supply, it might be rational and reasonable to postpone immunizing those people until we've gotten all the naive people who are negative, PCR negative or antibody negative, immunized, especially frontline workers. And so is the vaccine immunity durable? And the answer is we really don't know. I mean, this is a situation where it's clearly too soon to tell. We only have a relative limited period of time with people who have been provided with the vaccine, who we know have antibody present, neutralizing antibody and T cells as well. We know those are there. How long that's going to last, frankly, we don't know. So we can be hopeful, and I am hopeful, but we don't know. And so, summary. We have just witnessed an amazing technological breakthrough, just in time for your Hanukkah Christmas present for 2020. The authorized vaccines that have come from the FDA and will be shipped to a hospital near you in the immediate future look to be safe and effective. When offered, you should definitely take the vaccine. Vaccines work. This particular vaccine has been now deployed in Great Britain for almost a month. And as you know, with the possible provision of a few allergic reactions, seems to be very well tolerated by the people at the National Health Service and elderly patients in the UK as well. I should mention, talking about elderly patients, that both these vaccines seem to cause a robust response in seniors, those over 55 in the Pfizer vaccine and those over 65 in the group that was studied by Moderna. So it looks like seniors are going to be protected by this as well. There are side effects, fatigue, fever, aches, especially after the second dose, and there will be mishaps. And I don't want people to become discouraged if there are intermittent problems. And I'll just remind you that this has been the case with vaccine deployment over history. Now, we've had fits and starts. Those of you who are pediatricians, family practitioners, remember the stories of the rotavirus that didn't do particularly well, caused some problems when it was widely deployed. Also, some of you may remember or have read about the polio problems in the very beginning when one manufacturer failed to kill completely the virus that they were putting in the vaccine. There have been problems. We will have problems. This is not a perfect system, but I would urge you to keep the faith. This is a great advancement. These are good vaccines. They are effective and safe. And I want to make sure that you encourage yourself, your family members, your patients, everybody in your circle to take the vaccine when it becomes available. And please be careful. Let me re-emphasize and underscore the routine behaviors that have been successful so far in saving many of our lives. Hand washing, masking, social distancing, not getting into clusters and crowds, being, limiting your outside activity. These things are really, really important and can save your life and the life of seniors in your family. So that concludes this, and I would then entertain easy questions now. Emily, I would be happy to try.
Thank you, Dr. Pratt. That was wonderful. Go ahead
The
with
people
questions,
that are
please.
joining us on the WebEx system can unmute themselves to ask a question. So if you're able to see the screen, you can unmute yourself if you have a question.
David, this is John Assini.
Hi, John.
David,
Yeah.
question about numbers of patients that were studied with autoimmune diseases, since I'm certainly getting a lot of questions from primary care and other people about that. In other words, people with lupus and other things.
Right.
Any caveats as far as differences, or is basically what you said the only contraindication, the allergy to vaccine components?
So it seems, John, in the applications of both Pfizer and Moderna, they report immunizing people with malignancies and also with immune, abnormalities of one kind or another, autoimmune diseases and other things with very low frequency of problems. The other group, John, now that we're talking about special populations, is pregnant women. And this is a non-replicating vaccine. And the CDC currently recommends that if a woman expecting a child would like to be immunized, that we should go ahead and do that.
Great, thank you.
Hi, Dave. Roger Berlman. Great talk, really spectacular talk. Thank you. Question regarding, you had mentioned that there's suspicion that the number of cases that are present in the community are up to possibly 10 times the number that actually identified, meaning that some number of probably folks that are asymptomatic or undiagnosed, unreported. And I suspect that there is a huge amount to it. What evidence is there that the number is that high?
Yeah.
Given that there's 17 million cases in the U.S., that would mean half the U.S. in theory has already had it, and that's
Roger. I think probably it's closer to 2x.
Okay.
In the initial going, I thought it was a lot higher. We, remember that when they say the city of Albany has a background rate of 5% or 5.3 or 6%. That's not a random sample. That's people who decide to show up to be tested. So we get those numbers, and they're important and they're helpful. The real way to know, Roger, would be by doing large-scale seroprevalence. And seroprevalence goes out and selects a random sample of the American population, draws their blood, and looks for the presence of antibody. And that's really the way we'll get to know probably what the impact was. The state of New York did some early seroprevalence, and it was kind of all over the place. It was sky high in the boroughs and very low upstate. But eventually, as we do more and more deployment, these kinds of seroprevalence studies are going to have to be done because we need that. We need to know that data. But your question is a good one, and I'm just guessing. I'm guessing from my background in epidemiology to know that when you begin to see an outbreak and you see reported cases, it's almost always an underestimate. We're going to get closer to the real estimate as time goes by. But for now, I think certainly 17 million is an underestimate. I don't know what the right number is, but I am sure that's an underestimate.
Thank you.
Good morning. This is Larry Rautenberg, retired anesthesia. The CDC told us the past few years that we can no longer use multi-dose vials for more than one patient. It's got to be disposed of after the first patient, unless it's drawn up under a hood by the pharmacist away from patient areas. I have no problem with Ellis distributing it this way. They have the capability. I don't know if they have the manpower yet, but I have no confidence in our CVS's or Walgreens having that capabiliity. I have a couple levels going there.
Yeah, I can only say that think about how we get influenza vaccine. We get a unit dose preloaded in the syringe. You give it, right? So multi-dose vials everybody who's ever run an immunization program hates multi-dose vials for a million reasons, right? The only benefit of the multi-dose vials this time around is that there is more doses than we were promised by 20%. But I'm not sure. I don't know. Somebody from the pharmacy at Ellis might want to comment. But I think right now we're probably drawing them up and diluting them in the pharmacy. But I don't know about under a hood. But maybe one of the clinical pharmacists would comment.
Hi, this is Mike.
Oh,
Hello?
yeah, Mike Pasquarelli, go ahead.
Yeah, so this, and, Dr. Rautenberg, to his point, through P&T and whatnot, many times over the years. Yes, we're diluting them in the IV room, but the doses are actually being drawn up at the point of administration for the Pfizer product. And it'll be the same situation with the Moderna product, minus the dilution step, because it's already ready to go. I think the CDC has their recommendations with the multi-dose vials. I think the big issue here with regards to the retail settings is that there's one regulatory body that really doesn't have any oversight over the retail setting, and that's the Joint Commission. So they're the ones that generally tend to rear this issue to the forefront with the hospitals, but yet the retail setting gets a hall pass. So yes, there's valid concerns. I have yet to ever see there be or hear of any major issues. I'm sure there's issues with potential for contamination, but I'm sure it's very rare, if at all.
Thanks, Mike. Emily, I've got time for one more question.
Doc, can I ask a
I
question?
have a question from Dr. Lemire. He's been trying to unmute himself, but I have him on speakerphone.
Hi, Dave. Can you hear me okay?
I can.
Okay, anyway, number one, great talk, as usual. Two questions. The first one is, with the development of this vaccine and the way this was done by both these companies, do you think that going forward, for whatever we need a vaccine for next time, that this will have an impact on the whole process from beginning to approval, if that may speed the whole thing up? The second question is, when you answered the question of permanent immunity after infection, at least most of the stuff I have read, and maybe it deals with a subset of milder disease, is that antibodies last about 2 months. And maybe in people who were really, really sick, they might have longer protection, if you can comment on that. Thanks.
So, second question first. That is true. The data that the Chinese did substantial serology testing on people in recovery, and it did appear that the antibodies did begin to wane, probably 60 to 90 days. On the other hand, it doesn't mean that if you challenge them, they wouldn't have smart T cells, or they couldn't replicate immunoglobulins if you provoke them. So that's just one comment. But you're right. Your observation is correct. The second one is, will this technology be a template for what we do in the future, and will this kind of speed be likely in the future? And I think the answer to the first part is yes, and the second part is I'm not sure. I think the FDA and probably all of us would prefer the rigorous, more lengthy process if it's not a crisis like the one we're in right now. So those are my answers, the best I can do. But thank you for the questions. One more, Emily, then I really do have to go.
Yeah, Doc, this is Mike Pasquarello from the pharmacy. The question: any reputable reports of this current virus mutating?
Yes. There is a mutation that is being circulating in the United Kingdom. Someone needs to turn off their microphone. There is circulating virus that's mutated in the United Kingdom, and there are small mutations that are being isolated elsewhere. Now, it turns out that the coronavirus family is way more stable than in the RNA nature. The mRNA nature, not the RNA nature of this virus, is more stable than influenza. So the tendency, the proclivity toward mutation is considerably less in the coronaviruses than it is with the influenza family. So yes, the clinical implications of the mutation that has been isolated in the UK doesn't appear to be profoundly different, but there is mutation going on. Yep. So let me just thank everybody for joining me today. Wish you happy holidays, and please, 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 use the measures that we know that can save lives.
Thank you so much, Dr. Pratt. And please, if you need to leave, that's fine. The one other thing I really wanted to mention was the V-Safe system, which is after you get the vaccine, to sign up on your cell phone this system that has been developed by CDC. So we can all be part of what we'll call the phase 4, where we can easily report if we have any side effects. And again, that helps us all if we would join up with that system. Thank you again, everyone.
Right. Okay. Bye-bye.
Okay. Be safe. Take care.
Thank
And
you.
have
Bye-bye.
wonderful holidays.